উফ, Sure, last class we discussed about the translation process. Generally, the translation is the protein synthesis in the ribosome, and the main last class we discussed about the various component is required for the protein synthesis. First, the mRNA, which is the messenger RNA, or which type of protein is synthesized. Next one is the tRNA, or the truck that carry the amino acid and attach with the mRNA. And next which is ribosomal RNA, which is essential for the initiation of the or binding with the ribosome. Another is the ribosome, which is the factory house for the protein synthesis. Another is the aminoacyl-tRNA synthetase, which is essential for the amino acid binding, and the peptidal transferase, which is synthesis for the protein. And another is the GTP and GDP, which is the essential component for the energy supply, and another some protein factor, which is essential for the initiation of the protein synthesis. So next we can start what are the procedure for the translation of the protein synthesis. Generally the three procedure: one is the initiation, elongation, and the termination. How it will be formed? It will be generally formed in the ribosome in case of prokaryotic as well as the eukaryotics. In case of eukaryotics is the 80s ribosome, in case of prokaryotic 50s, but having the two subunit, one the small, another the large subunit. In case of prokaryotic 70s, the large subunit is the 50s and small unit is the 30s. In case of eukaryotics 80s, their large unit is the 60s and the small unit is the 40s. After all, it will be formation of protein synthesis. The translation of RNA direct the synthesis for the polypeptides, and translation involve mRNA, ribosome, transfer RNA, and genetic code. Obviously, the genetic code I already discussed about the details about the genetic code. This is the codon, and these are the anticodon, the gene 1, gene 2, gene 3. The genetic code, last class we discussed about the genetic code, characteristic of the genetic code, transfer RNA. Next, we go to the transfer RNA. It is considered a single RNA strand that is only about the 8 nucleotides long, and each carries a specific amino acid one end to another end and has a anticodon, and on the other hand, the special group of enzymes is paired to the proper tRNA molecules within the corresponding amino acid. This is very important. This anticodon part which recognize the mRNA, and this 3' end which is the recognize the specific amino acid, and 5' end which produce the peptidal chain or the protein. The special group of enzymes pair up with the proper tRNA molecules with their corresponding amino acid. This tRNA binding the amino acid to the ribosomes. This anticodon in the 3 RNA base that are matched to the 3 base of the codon on the mRNA molecule. It is the two-dimensional structure. 1 minute. हेलो हेलो हाँ बोलो हम्म 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 তো জিজ্ঞেস করতে পারেন ঠিক আছে, ওকে জিজ্ঞাসা করো, কিছু অসুবিধা নেই, আমার ইয়ে আছে, পরিচিত আছে ভালো। মানে যাই একটু আরেকটু স্যার, দাদা একটু ভুলে গিয়েছিলাম, বা স্যার যাই বলো যে ভুলে গিয়েছিলাম কোন ডিভিশন, কারণ ও ডিভিশনটা তো লিখে নি, আমিও খেয়াল করে নি। না না না, ঠিক আছে, কথা বলে না, আমি ক্লাসে আছি, কিছু অসুবিধা নেই, ধরবে ও। ঠিক আছে। These are the two-dimensional structure. The four base pairs regions and the three loops are the characteristics of the all tRNA. All tRNA. And as the base sequence of the amino acid attachment at the 3 end, be careful, always remember it, the amino acid attached in the 3 end and the anticodon triplet is unique to each tRNA because this tRNA anticodon which bind with the codon part of the mRNA. These are strict parts of this. This is very important, is labeled the chemically modified and characteristics tRNA. The tRNA, the three-dimensional structure of tRNA, it is roughly L in shape. The amino acid attachment site in the 3' end and peptide attachment all are the hydrogen bonding and anticodon part which is attached with the codon part of the mRNA. The ribosome, which is the practical house for the protein synthesis, ribosome family, facilitate the specific coupling of the tRNA anticodon with the mRNA, which all this coupling process occur in the ribosome and during the protein synthesis. And the two ribosomal subunits are constructed the protein and mRNA molecule, RNA molecules named ribosomal RNA or RNA. These are the how it will be translated. Here it is the DNA, mRNA, ribosome, translation, and peptide will be formed. This is the exit unit for the growing the polypeptides EPA. One is the E site and the P site and here A site. A site for the amino acid binding site, P for the polypeptide, and it is the exit site. And the large subunit, one is the small subunit. And the compartmental model of the function of this, it is the model of bacterial ribosome showing the overall shape. And this, in case of eukaryotic ribosome, it is roughly similar, but a ribosomal subunit is aggregates of a ribosomal RNA molecule and the proteins. These are the ribosome structure. These are the ribosome, and these ribosome have the three binding site. One is the A site and the P site and the E site. P site for the peptidal tRNA binding site or the formation of the peptide bond or the formation of protein. A site is the aminoacyl-tRNA synthase where the amino specific amino acid bind to each other, and E is the exit site. And the one is the large subunit, another the small subunit. It is a schematic model shows the binding. The site of the ribosomes has been mRNA binding and three tRNA binding site and known as the APE sites, and this schematic ribosome will appear in the later diagram. This building a polypeptides number the amino acid by amino acid N and growing polypeptide and the next amino acid chain and ultimately the mRNA and the codon is formed. So schematic model with the mRNA and tRNA, this tRNA will be fit binding with the site of the anticodon base pair with the mRNA codon. The P site will be hold the tRNA attached with the growing polypeptides and a site of whole tRNA carrying the next amino acid to add it to each others, and finally the discharge the tRNA and leave via the exit site. And binding a model of the tRNA, the specific enzymes called the aminoacyl-tRNA synthase join each other amino acid to the correct tRNA. This is also very important factors. The amino acid bind with the tRNA, that particular tRNA which bind with the specific anticodon, and that will bind with the codon part of the mRNA. The amino acid tRNA site is an active site binding for the amino acid and which is ATP dependent process. The amino acid adenosynthase, and number two, the ATP losses two P groups up and joining amino acid and the phosphorylate groups of the phosphate 3 appropriate tRNA covalently bond to amino acid and displacing the amino acid. হ্যাঁ বলো 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 বলে যাও তাড়াতাড়ি না না 10 হয়ে যাবে না না 10টা তো 21 হয়ে চলে গেছে হ্যাঁ তুমি 10ই থাকো তো বলে দিয়েছিলাম তো ঠিক আছে আমি বলে দেবো এখন বলো 10 নামটা তো দরকার ঠিক আছে হুম 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 Next, number 3, this is the approximate tRNA and covalently bond to amino acid and displacing the amino acid. Activation of amino acid is released by the enzymes aminoacyl-tRNA, or is the activation amino acid, the figure. So amino acid bind with this to the tRNA, build a polypeptides. We can divide the translation into 3 stages. One is the initiation, elongation, and the termination. The AUG, it is a start code. It is recognized by the methionine tRNA or MET tRNA. Once the transcodon has been identified, the ribosome incorporates an amino acid into the polypeptide chain, and RNA is decoded by the RNA or transfer RNA molecules. And which each transport specific amino acid to be growing chains, and translocation ends when a stop codon UAG and UGA is reached. So first of all, the initiation. How to initiation? The AUG is the starting codon or the methionine or MET, and then it is to be elongated. One amino acid attached to the another acid, then amino acid. It is depend upon the code of the mRNA. Suppose mRNA code contain 50 codon, so at least 50 per amino acid bind sequentially. After that it will be completed, and after that one stop codon will come out of 3 stop codon, then immediately stop. So 50 amino acid containing sequentially 50 amino acid containing protein will become. The initiation of the translation, the initiation stage of the translation bring together mRNA, tRNA, and the first amino acid of the polypeptide and 2 subunit of the sub enzymes in the first codon. This initiation of the tRNA, here it is a codon, this anticodon bind with this the MET tRNA, this is the start codon and small ribosomal and translation. That means one amino acid bind with the anticodon part of the specific amino acid, then it will be released by the exit point, then another tRNA will become, then another, and finally the elongation will be completed, and ultimately the total length of the amino acid sequence will be formed, and that will be joined by the amino peptidal transferase by the one amino acid bind with the another amino acid by the peptidal transferase or by the formation of the peptide bond. So elongation of the peptide chain, the elongation stage of the amino acid are added to by on the preceding the amino acid. You can see it. The first one is the amino acid peptidal transferase. Here the exit side, this is the amino acid mRNA sequence. In that sequence, the anticodon is bind with this, the codon recognition. The anticodon of an incoming aminoacyl-tRNA base pair with the complementary mRNA codon in the A site. And hydrolysis of GTP increase the accuracy and efficiency step. Number 2, the GTP to GDP is convert. Next, first amino tRNA will be released for the exit point, and then the peptide bond will be formed. A tRNA molecule of the large subunit catalyze the formation of a peptide bond between the new amino acid in the A site and the carboxylic end of growing polypeptide in the P site. The steps attached the polypeptide to the tRNA A site. Next is the pRNA site, the translocation, that means transfer, then one tRNA, then another tRNA containing amino acid. That way ultimately the peptide will be growing. These are the according chain length of the protein chain will be increases. So the empty tRNA of P site will be removed and exit site where it is be released, and mRNA removed along the bound of the tRNA and bring a next codon to be translated of the A site. So finally the complete sequence of amino acid will be formed after the sequence as per the mRNA. Termination, how it will be terminated? It is not continuous process, it will be terminated. Every process is terminated. How it will be terminated? It will terminated by the when the stop codon is come out of 3 stop codon, this final step of the translation is the termination. And when the ribosome reach as stop codon, there is a no corresponding transfer of tRNA, and instead a small protein called the release factors, and these attach to the stop codon, and these release factor cause whole the complex to fall apart, and messenger RNA, the two ribosomal subunit, the new polypeptides, and this messenger mRNA are translated many times to produce many protein copies and release. Ultimately, the stop codon will become, after that, the stop codon will because to inhibit the protein synthesis, and ultimately they stop the protein synthesis. When the ribosome reaches to the stop codon of even A site, ribosome accept the protein is called and release the factor, some releasing factor, and this release factor hydrolyze to bond between the tRNA in the P site, because tRNA of the P site, that means the tRNA will not work at last amino acid of polypeptide chain, and this polypeptide is the speed from the ribosome, and the two ribosomal subunit and the other components of the assemble and dissociated. Translation or initiation in this mRNA ribosome and transfer tRNA corresponding to the start codon binding this complex, and some ribosome are complex of the two subunit large and small which come together, and when a messenger RNA attached to the DNA its translation ribosome tRNA will become, this is the mRNA and this is the tRNA, and met tRNA, this ribosome one tRNA then another tRNA then another tRNA as per the codon and anticodon binding specification. Ultimately a protein will be synthesized. Translation or elongation, there is a two. So here is the P site, the metformin leucine, there are 1 2, these are the two bond. Ultimately as per the sequence of the codon of the mRNA, there is a number of tRNA will become, and finally the stop codon will become, and ultimately the tRNA bind with this, and to prevent the to stop the protein synthesis. Polyribosomes. A number of ribosomes can translate the single mRNA molecule and simultaneously forming the polyribosomes. That means not a single ribosome, but the simultaneously protein synthesis occur in the different ribosomes, and thus polyribosomes enable to cell to make a many copies of the polypeptide. It's a very quickly. So this is called the polyribosomes. And combining the gene expression in the prokaryotics and the eukaryotics, it will be discussed in next class, gene expression. And before that, we discuss about the what are the post translation modification of the protein synthesis. Post translation modification, these are the post translation modification of the protein synthesis. Next class we discuss about it. Okay, thank you very much for listening the lecture. If any question, please ask me freely.